Number 1. The pregnant Plesiosaur, frozen mid-gestation. 78 million years ago, a shallow sea split North America in half. The western interior seaway stretched from the Gulf of Mexico to the Arctic, 200 to 300 meters deep, warm, and teeming with life. Mosasaurs cruised the upper waters. Sharks patrolled the thermocline. And among them, a 4.7-meter polycotyledonid Plesiosaur, short-necked, barrel-bodied, built for speed, was carrying something no paleontologist would confirm for another 131 million years. In 1987, a rancher named Charles Bonner found the skeleton eroding out of Pierre Shale on his property in Logan County, Kansas. The specimen was collected by the Natural History Museum of Los Angeles County and cataloged as LACM 129639. For over 2 decades, it sat in the collections as a well-preserved but unremarkable polycotylid. Then, in 2011, paleontologists F. Robin O'Keefe and Luis M. Chiappe published a paper in the journal Science that changed everything we thought we knew about how these animals reproduced. Nestled between the mother's pelvic and pectoral girdles was a second skeleton, a fetus, approximately 1.5 meters long, roughly 1/3 the length of the adult. CT scanning confirmed this was not a prey item. The bone density was wrong. The orientation was wrong. The fetal bones showed incomplete ossification in the vertebral centra and limb elements, exactly what you would expect from a developing embryo. And the fetus was positioned head toward tail, the same orientation seen in modern dolphin and whale births, where tail-first delivery prevents the newborn from drowning before it reaches the surface. This was the first direct fossil evidence that plesiosaurs gave live birth, not on a beach, not in a nest, in open water. O'Keefe and Chiappe went further. A single large fetus, 1/3 the mother's body length, pointed to what biologists call K-selected reproduction. Instead of producing dozens of small offspring and abandoning them to chance, this animal was investing everything in one. The implication was startling. If plesiosaurs bore single, large young, they almost certainly provided parental care. They may have nursed, they may have guarded, they may have behaved in broad strokes like modern whales. Not everyone was convinced. Michael Caldwell at the University of Alberta cautioned that a sample size of 1 limits how far you can generalize across an entire order of marine reptiles. Plesiosaurs spanned over 135 million years and dozens of families. Whether long-necked elasmosaurids and giant pliosaurs reproduce the same way remains unknown. Adam Smith at the University of Nottingham argued that the body plan of long-necked plesiosaurs, with flippers instead of legs and no ability to support their weight on land, made egg-laying physically implausible. But implausible is not impossible. The ichthyosaurs of Holzmaden, Germany, had already proven viviparity in marine reptiles, with exquisitely preserved specimens showing embryos inside the mother. What Polycotylus added was the suggestion that marine reptile reproduction could be not just viviparous, but mammalian in its investment. The specimen is currently on display at the Natural History Museum of Los Angeles County. A mother and her unborn child, frozen in stone, 78 million years into a birth that never happened. Number 2. Bloodfalls. There's a place in East Antarctica where a glacier bleeds. At the snout of Taylor Glacier, in the McMurdo dry valleys of Victoria Land, a 5-story cascade of crimson liquid pours from a crack in the ice face and stains the surrounding snow the color of an open wound. It has been doing this, intermittently, for as long as anyone has been watching. The first person to describe it was the geologist Griffith Taylor during Robert Falcon Scott's 1911 Terra Nova expedition. Taylor assumed the red color came from algae. He was wrong. The red is iron. Specifically, it is hypersaline water saturated with ferrous iron, iron in its reduced dissolved form that has been sealed beneath the glacier for approximately 1.5 to 2 million years. When this ancient brine finally reaches the surface and contacts atmospheric oxygen, the ferrous iron oxidizes to ferric iron, and the water turns the color of blood. But the iron is not the discovery. The discovery is what lives in it. In 2009, geomicrobiologist Jill McCookie at the University of Tennessee published a paper in Science that identified an active microbial community thriving in this brine. at least 17 distinct microbial taxa, including members of Theomicrospira and Disulfocapsa, surviving in water 3 to 4 times saltier than the ocean, at a temperature of negative 7 degrees Celsius, with zero dissolved oxygen in total darkness, for over 1 million years. How? McCookie's team used isotopic analysis to unravel the metabolic pathway, and what they found had never been documented anywhere on Earth. The microbes were using sulfate as a catalytic electron shuttle to reduce ferric iron for energy. They had built an entire biochemistry around the chemistry of iron and sulfur, without a single photon of sunlight. The brine itself originates from an ancient fjord, a body of seawater that was trapped and sealed when Taylor Glacier advanced over it during the Neogene. The organisms living in it descend from marine camellithotrophic lineages that were present in that original fjord water. They have been evolving in isolation, in darkness, under 400 meters of ice, while the entire genus Homo appeared and spread across the planet above them. In 2015, Aaron Pettit and colleagues at the University of Alaska Fairbanks used radio echo sounding to map the hydraulic pathway. The brine travels approximately 4 kilometers through fissures and cracks in Taylor Glacier before emerging at the surface. The subglacial reservoir feeding Blood Falls has never been directly sampled. Everything we know comes from the outflow. The salinity is approximately 120 grams per liter of dissolved solids compared to 35 for normal seawater. The iron concentration is approximately 0.3 millimoles per liter, extraordinarily high for any natural water body. The dry valleys surrounding the site are considered the most Mars-like environment on Earth. Average temperatures of negative 20 degrees Celsius, annual precipitation below 50 millimeters, and katabatic winds exceeding 320 kilometers per hour. NASA and the National Science Foundation have funded research at Blood Falls since the early 2000s as part of astrobiology programs. The logic is straightforward. In 2018, the MARSI radar instrument on the European Space Agency's Mars Express orbiter detected what appeared to be subglacial brines beneath Mars's south polar ice cap. Jupiter's moon Europa has a subsurface ocean beneath an ice shell estimated at 10 to 30 kilometers thick. If life can persist for 1 million years in a frozen, oxygen-free, sunlight-free brine beneath an Antarctic glacier, the universe of possible habitats just expanded enormously. The researchers from the SALSA program, Subglacial Antarctic Lake Scientific Access, have used Blood Falls as a testing ground for clean access drilling technologies designed to sample these environments without contaminating them. The stakes are high. If we find life beneath the ice of another world, the first question will be whether we brought it with us. Number 3, the Mosasaur Cannibalism Graveyard. The Niobrara Formation of western Kansas is a layer of chalk deposited on the floor of a shallow sea between roughly 87 and 82 million years ago. It is, by specimen count, the richest Mosasaur locality on Earth. Thousands of fossils have been pulled from this chalk since the 1870s, many by the legendary Sternberg family, who spent decades crawling across the Kansas Badlands with chisels and plaster. What they found was a killing field. Mosasaurs were the apex marine predators of the late Cretaceous. The largest species, Mosasaurus hoffmanni, reached 17 meters in length. At least 6 genera coexisted in the Niobrara Seaway: Tylosaurus, Cladastes, Platycarpus, Plioplatycarpus, Halosaurus, and Mosasaurus, occupying overlapping niches in a body of water barely 200 meters deep. The ecosystem was oversaturated with large predators. And large predators, when they run out of easier options, eat each other. In 2004, Michael Everhart at the Sternberg Museum of Natural History documented bite marks on a Tylosaurus proriger specimen, designated FHSMVP-3, that matched the tooth spacing and morphology of another, larger Tylosaurus. Some of the bite marks showed signs of healing, meaning the animal had survived at least one attack from a member of its own species before eventually dying. Everhart went on to catalog at least a dozen specimens from the Smoky Hill Chalk member, showing clear evidence of Mosasaur on Mosasaur predation. In 2009, Johan Lindgren described a specimen even more explicit: a juvenile Cladastes leodontis preserved inside the stomach cavity of a larger Tylosaurus proriger, cataloged as LACM128319. The juvenile had been swallowed largely intact. Its vertebral column was still articulated within the gut region of the predator. This was not scavenging. This was a large mosasaur hunting and consuming a smaller one whole. Isotopic analysis by Clements and colleagues in 2014 confirmed the hierarchy chemically. Carbon and nitrogen isotope ratios from mosasaur teeth showed that larger species occupied a higher trophic level than smaller species, the isotopic signature of intragill predation. Tylosaurus ate cladastes. Cladastes ate fish and ammonites. The food web was structured by size, and it was brutal. Takuya Konishi, then at the University of Cincinnati, described a plate carpus specimen bearing both a shark tooth embedded in its vertebra and mosasaur bite marks on its paddle. Two different predators had targeted the same individual, possibly in rapid succession. Pathological analysis of bones from Kansas and South Dakota revealed healed bite wounds consistent with conspecific combat, fights between members of the same species that the loser survived, at least temporarily. Poulton and colleagues proposed in 2013 that mosasaur cannibalism may have intensified during periods of declining prey availability in the final 2 million years of the Cretaceous. The dental morphology tells its own story. Globidins had rounded, crushing teeth built for shellfish. Tylosaurus had conical, piercing teeth designed for one thing, killing large prey, including other mosasaurs. The parallel to modern marine ecosystems is striking. Great white sharks and orcas both display intragill predation and occasional cannibalism, particularly when resources are constrained. The pattern appears to be universal among apex marine predators across hundreds of millions of years. When you are at the top of the food chain and the food chain contracts, you eat what is left. And what is left is each other. The mosasaurs left no descendants. Every lineage was extinguished at the Chicxulub impact 66 million years ago. What remains is the chalk of Kansas, packed with the evidence of a predatory arms race that ended only when an asteroid the size of a city made the question irrelevant. Number 4. The Lake Vostok organisms. Beneath Russia's Vostok station, at 78 degrees 28 minutes south latitude, 106 degrees 48 minutes east longitude, there is a body of water the size of Lake Ontario that has not seen sunlight in at least 15 million years. Lake Vostok is the largest known subglacial lake in Antarctica, roughly 250 kilometers long by 50 kilometers wide, with a surface area of approximately 12,500 square kilometers and a maximum depth approaching 1,000 meters. It sits beneath 3,769 meters of ice. The ice above Vostok is itself a scientific treasure. The ice core extracted from this site, published by Petit and colleagues in Nature in 1999, recorded 420,000 years of continuous climate history, the longest unbroken atmospheric record ever recovered. But the real question was always about what lay below the ice, in the water. Russian drilling at Vostok began in 1989. It took 23 years. On February 5, 2012, the drill reached the ice-water interface at a depth of 3,769.3 meters. They had punched through to a lake that had been sealed from the atmosphere since the East Antarctic ice sheet first formed over this region, sometime between 15 and 25 million years ago. What they found was controversial from the start. The accretion ice, the layer of refrozen lake water in the lowest 200 meters of the core, contained DNA. Scott Rogers and colleagues at Bowling Green State University published their findings in PLOS One in 2013, identifying at least 3,507 unique genetic sequences. The organisms included bacteria, both thermophilic species adapted to heat and psychrophilic species adapted to cold, along with fungi and sequences matching freshwater crustaceans and rotifers. Some of the bacterial sequences match species typically found in deep-sea hydrothermal vents, implying geothermal activity on the lake floor. A 2016 study by Starkman and colleagues pushed the findings further, identifying sequences matching fish parasites of the genus Diphyllobothrium and thermophilic bacteria of the genus Hydrogenophilus. If genuine, this implied an ecosystem far more complex than a few extremophile bacteria, potentially a food web with multiple trophic levels sustained entirely by geochemical energy from the bedrock. But the contamination debate has never been resolved. Sergei Bulat at the Petersburg Nuclear Physics Institute reported finding extremely low concentrations of microbial cells, only about 280 per milliliter, in the accretion ice. The Russian drilling program used kerosene and freon as drilling fluids. Distinguishing native organisms from contaminants introduced during 23 years of drilling has proven extraordinarily difficult. The lake itself is divided into two basins by an underwater ridge. The northern basin is approximately 400 meters deep. The southern basin approaches 1,000 meters. The water temperature hovers around -3 degrees Celsius, kept liquid by the immense pressure, approximately 350 atmospheres of the overlying ice sheet. And the dissolved oxygen concentration is estimated at 50 times higher than typical surface freshwater, supersaturated by oxygen released from the melting base of the ice. At those concentrations, oxygen becomes toxic to most known organisms. John Priskew at Montana State University, one of the leading scientists in Antarctic subglacial lake research, has argued that the low but consistent cell counts represent a genuine subglacial biosphere sustained by geochemical energy. Others remain skeptical. The British-led Lake Ellsworth drilling project in 2012 and the American SALSA project at Lake Mercer in 2018 and 19 attempted clean access drilling to avoid the contamination issues that plagued Vostok. Their results confirmed microbial life in subglacial water, but at other lakes, not Vostok itself. If the Lake Vostok ecosystem is confirmed as genuine, it would represent the most isolated biosphere on Earth and the most direct analog for potential life beneath the ice shells of Europa and Enceladus. NASA's Europa Clipper mission, launched in October 2024, will investigate conditions on that moon, partly informed by what we have learned and what we have argued about in this sealed, pressurized, oxygen-saturated lake beneath the coldest place on the planet's surface. Number 5. The giant sea spiders. There are creatures walking across the floor of the Southern Ocean that look like they were designed to be nightmares. They are sea spiders, class Pycnogonida, and in Antarctic waters, they grow to sizes that defy everything you think you know about arthropod body plans. Leg spans exceeding 70 centimeters, 8 legs in most species, but 10 in Decalopodi australis, bodies so reduced that their gut extends branching diverticula into every single leg because the central body is too small to contain the digestive system. They digest their food throughout their entire body, and they move across the seafloor at the pace of geological time, inserting a long proboscis into soft-bodied prey, hydroids, bryozoans, anemones, nudibranchs, and sucking them dry from the inside. The phenomenon is called polar gigantism. Organisms in cold polar waters grow to sizes far exceeding their temperate and tropical relatives. The most common Antarctic species, Colocendeus megalonyx, reaches leg spans of 50 to 70 centimeters. In tropical waters, the same class of animals rarely exceeds 1 to 2 centimeters. The leading explanation was proposed by Art Woods at the University of Montana and colleagues in a 2009 paper in the Proceedings of the Royal Society B. Cold water holds more dissolved oxygen. Pycnogonids have no dedicated respiratory organs. Gas exchange occurs entirely across the surface of their exoskeleton and through the thin walls of their legs. In warm water, where oxygen is scarce, this limits body size. In the oxygen-saturated waters of the Southern Ocean, the constraint lifts. Woods' team experimentally demonstrated that pycnogonid metabolic rates scale with temperature and that larger specimens require the high oxygen conditions found only at the poles. A 2017 follow-up study by Caitlin Shishido and Woods showed that larger pycnogonids have proportionally thinner cuticles, allowing more efficient oxygen diffusion to sustain their oversized bodies. The Southern Ocean has been thermally isolated by the Antarctic circumpolar current for approximately 34 million years since the opening of the Drake Passage in the late Eocene. That isolation has produced extreme biological endemism. An estimated 50 to 70% of Antarctic marine invertebrate species are found nowhere else on Earth. Giant isopods, giant amphipods, giant ribbon worms, polar gigantism is not limited to sea spiders. It is a systematic feature of an ecosystem that has been sealed in cold, oxygen-rich water for longer than most mammal lineages have existed. Pycnogonids themselves are ancient. The oldest known fossil from the Orsten fauna of Sweden dates to approximately 500 million years ago, making them one of the most ancient arthropod lineages on Earth, molecular phylogenetics suggests the Antarctic radiation diversified primarily during the Miocene, coinciding with the intensification of Antarctic glaciation and the thermal isolation that made gigantism possible. But polar gigantism is fragile. Stephen Chown at Monash University and colleagues have documented the thermal tolerance limits of Antarctic marine invertebrates. Many cannot survive temperature increases of just 2 to 3 degrees Celsius above current ambient conditions. The Southern Ocean has warmed by approximately 1 degree Celsius in the upper 500 meters since the 1950s. If that trend continues, the dissolved oxygen concentrations that sustain these oversized bodies will drop. The males who carry thousands of fertilized eggs on specialized ovigerous legs for months at a time will be the first to feel the metabolic squeeze. The giant sea spiders of Antarctica are not relics of a past era. They are products of a very specific set of conditions, cold, oxygen, isolation, time, that are now changing faster than at any point in the 34 million years since the Drake Passage opened. Number 6. The standing petrified forests. In the Transantarctic Mountains, at elevations exceeding 2,000 meters, there are tree stumps still standing where they grew 260 million years ago. They have not fallen. They have not been transported. They are rooted in the same Plesiosaur, the ancient soil, where their seeds first germinated in the Permian, on a continent that was then part of the southern supercontinent Gondwana, and positioned at a paleolatitude of roughly 60 to 70 degrees south. The trees are Glossopteris, the dominant genus of the southern hemisphere Permian. A deciduous seed fern with distinctive tongue-shaped leaves that would later become one of the most important pieces of evidence for continental drift. Eduard Suess identified the genus in 1861. Alfred Wegener used its distribution across South America, Africa, India, Australia, and Antarctica to argue that these landmasses had once been joined. The trees were proof of a vanished world long before anyone drilled into the ice to confirm it. Eric Gulbrandsen at the University of Wisconsin-Milwaukee led expeditions to the Transantarctic Mountains in 2016 and 2017, recovering specimens that revealed an ecosystem far more complex than anyone expected. At a single site in the Allen Hills, Gulbrandsen's team identified at least 13 species of petrified trees. This was not a monoculture. It was a diverse forest, growing in months of continuous summer sunlight and enduring months of total polar darkness each winter. The growth rings tell the story. Analysis of the petrified wood shows extremely rapid growth during the polar summer. Some trees added 3 to 4 centimeters of wood in a single growing season compressed into just 4 to 5 months of continuous light. Then they shut down entirely for the winter. The deciduous strategy was not optional. It was the only way to survive at 60 to 70 degrees south latitude, where the sun does not rise for weeks on end. The preservation is extraordinary. The trees were silicified, their organic material replaced atom by atom with silica during burial, preserving cellular detail down to individual xylem cells, growth rings, and even fungal hyphae threading through the wood. Mycorrhizal fungi, the symbiotic root networks that help modern trees access nutrients, were present in the fossilized root systems, indicating complex soil ecosystems and nutrient cycling networks supporting the forests. These forests were destroyed during the most catastrophic event in the history of life on Earth, the end Permian extinction, approximately 252 million years ago, killed roughly 96% of all marine species and 70% of terrestrial vertebrate species. Volcanic ash layers interbedded with the fossil forest deposits link the tree death directly to the eruption of the Siberian Traps, the largest volcanic province ever documented, which erupted approximately 4 million cubic kilometers of lava over the span of roughly 1 million years. Some of the stumps show charring and charcoal, evidence that the forest burned as atmospheric temperatures spiked and oxygen levels crashed. But Antarctica's forests did not end with the Permian. Triassic forests followed, including some of the earliest known examples of modern conifer-like trees, documented by Patricia Ryberg at Park University. Cretaceous age forests, dating to roughly 90 to 100 million years ago, have been found in the Antarctic Peninsula region, including tropical and subtropical species, Araucaria, ferns, and early angiosperms. During the mid-Cretaceous, Antarctica's mean annual temperature was approximately 13 to 20 degrees Celsius. There was no permanent ice sheet. Forests extended to within 5 degrees of the South Pole. The transition from forested Antarctica to ice-covered Antarctica was not a single event. It occurred gradually between approximately 50 and 34 million years ago, driven by declining atmospheric carbon dioxide levels and the thermal isolation created when the Drake Passage opened and the Antarctic circumpolar current began to flow. The last forests on the continent, consisting of Nothofagus or southern beech and podocarps, held on until approximately 12 to 14 million years ago on the Antarctic Peninsula before the advancing ice sheets finally eliminated them. What stands in the Transantarctic Mountains today is the memory of a world where Antarctica was green, alive, and warm enough to grow trees at latitudes where nothing now survives but wind and ice. Number 7. The subglacial Lake Whillans microbiome. In January 2013, a team of scientists led by John Priscu at Montana State University and Sławek Tulaczyk at the University of California, Santa Cruz, drilled through 802 meters of West Antarctic ice and penetrated a body of water that changed the way we think about life on this planet. Subglacial Lake Whillans lies beneath the Whillans ice stream at 84 degrees 14 minutes south, 153 degrees 42 minutes west. It was first identified through satellite altimetry. Instruments detected the ice surface rising and falling by approximately half a meter as the lake filled and drained in cycles. The lake itself turned out to be almost absurdly shallow. The water column was only about 2.2 meters deep. A person could stand in it. But what the Wizard Project Whillans Ice Stream Subglacial Access research drilling found in that shallow water column was profound. The drilling used hot water sterilized with ultraviolet light, filtered, and pasteurized to prevent contamination, making this the first clean access penetration of a subglacial Antarctic lake, and the water they pulled up was not sterile. Brent Christner, then at Louisiana State University, led the microbial analysis, published in Nature in 2014. The water contained approximately 130,000 microbial cells per milliliter, orders of magnitude higher than the contested counts from Lake Vostok. And these were not dormant. They were metabolically active, they were growing, they were eating. The dominant organisms were chemolitho-autotrophic bacteria, organisms that derive energy not from sunlight, not from consuming other organisms, but from chemical reactions between water and the minerals in the rock beneath them. Nitrifying bacteria related to Nitrosospira and Nitrospira were among the most abundant, oxidizing ammonium from the underlying sediment for energy. Sulfur-oxidizing bacteria related to Thiobacillus and iron-oxidizing bacteria were also present. The community was fixing carbon, pulling CO2 out of the water, and converting it into organic carbon using chemical energy from geology. It was photosynthesis without the photo, a biosphere running on rock. Amanda Ochberger, also at Louisiana State University, published a detailed phylogenetic analysis in 2016 using 16S ribosomal RNA sequencing. She identified at least 3,900 operational taxonomic units, 3,900 distinct microbial types in a lake 2 meters deep under 800 meters of ice in permanent darkness. Metagenomic analysis revealed genes for nitrogen cycling, sulfur metabolism, and iron metabolism, a complete biogeochemical cycling community doing everything a surface ecosystem does without a single ray of light. The sediment at the bottom was clay-rich glacial till, ground-up rock left behind by the moving ice stream, laced with minerals that provided the chemical substrates the microbes needed. The water was slightly brackish, reflecting millions of years of rock-water interaction. Unlike the Lake Vostok, Lake Whillans is not a sealed system. It connects to a broader subglacial hydrological network that fills and drains on cycles of roughly 3 to 7 years. When the lake drains, it sends pulses of microbial-laden water to the grounding zone, where the Whillans ice stream meets the Ross Sea, potentially seeding the marine environment with organisms from beneath the ice. To date, over 400 subglacial lakes have been identified beneath the Antarctic ice sheet. Priscu has estimated that the total microbial biomass living beneath Antarctic ice could rival that of all Earth's freshwater lakes combined. An entire biosphere, as old as the ice sheet itself, running on chemistry and darkness, connected by rivers no one has ever seen. Number 8, the frozen whale barnacles. Every humpback whale that migrates through the Southern Ocean carries passengers. Whale barnacles, family Coronalidae, are obligate commensals. They live exclusively on the skin of large whales and cannot survive on any other surface. The most common genus, Coronulae, embeds directly into the whale's epidermis, individual specimens reaching 5 to 7 centimeters in diameter. They are born in the water column, settle on a passing whale as larvae, and spend their entire adult lives recording everything that whale does, everywhere it goes, in the chemistry of their shells. In 2019, Larry Taylor and colleagues published a paper in Nature Communications that demonstrated something remarkable. The oxygen isotope ratios, delta 18O, in whale barnacle growth layers recorded the temperature of the water through which the whale traveled. Each growth layer represents approximately 1 day of growth, creating a high-resolution diary of the whale's journey across different water temperatures and latitudes over the barnacle's 3 to 6 month lifespan. Taylor's team applied this technique to barnacle specimens recovered from Pleistocene deposits approximately 125,000 years old. By reading the isotopic record in each growth layer, they reconstructed humpback whale migration routes from the last interglacial period and compared them to the routes used by modern populations. The results were striking. The Pleistocene whales were following essentially the same paths. The migration patterns were not learned behavior, passed culturally from mother to calf. They were deeply conserved, programmed by hundreds of thousands, potentially millions, of years of evolutionary pressure. Whale barnacles evolved approximately 3 to 5 million years ago during the Pliocene, coinciding with the diversification of modern baleen whale lineages. Their shells are composed of calcite, calcium carbonate, which preserves exceptionally well in cold, deep marine sediments. Barnacle fragments recovered from International Ocean Discovery Program drill cores in Antarctic and sub-Antarctic waters extend the migration record back millions of years, offering a window into whale behavior that no other source can provide. The shells also record strontium isotope ratios, which can be used to determine the age of the deposits and the specific marine environments the whales occupied. Daniel Burgel and colleagues used lipid biomarkers within barnacle-associated sediments to reconstruct Southern Ocean productivity during glacial-interglacial cycles. The barnacles, without any agency of their own, were recording not just where whales went, but what the ocean was doing when they got there. The relationship between whale and barnacle is ancient and apparently coevolutionary. Fossil barnacles from Pliocene deposits in Peru and Chile show morphological differences from modern forms, suggesting both partners have been shaping each other for millions of years. Coronula diadema creates a distinctive rosette-shaped cavity in whale skin, and the scars left when barnacles detach can be used to identify individual whales, a natural tagging system that requires no human intervention. Industrial whaling nearly destroyed both partners. The near extinction of humpback whale populations before the 1966 moratorium devastated coronula populations alongside their hosts. Recovery has been slow. But in Antarctic marine sediments, in drill cores pulled from the seafloor alongside diatom and foraminiferal ooze, the barnacle record persists, an unbroken archive of whale movement, ocean temperature, and marine productivity stretching back into deep time, written in calcium carbonate by organisms that had no idea they were keeping records. Number 9, the Gamburtsev Ghost Mountains. There is a mountain range the size of the European Alps buried beneath the East Antarctic ice sheet, and no human being has ever seen it. The Gamburtsev subglacial mountains extend roughly 1,200 kilometers beneath the ice, centered at approximately 80 degrees south, 76 degrees east, with peaks reaching 2,700 meters above sea level. They have sharp, rugged summits, deep U-shaped valleys. The kind of topography you would expect from the Swiss Alps or the Rockies, and they are covered by more than 3 kilometers of ice. They were first detected in 1958 by Soviet geophysicists using seismic sounding during the International Geophysical Year. They were named after Grigory Gamburtsev, the Soviet geophysicist who pioneered the deep seismic methods that made their detection possible. For 50 years, they remained little more than a blip on a seismograph, evidence of something massive hiding beneath the ice, but with no detail. That changed between 2008 and 2009, when the AGAP survey, Antarctica's Gamburtsev province, deployed airborne radar, magnetics, and gravity instruments across the region in a 7-nation international collaboration. The project was led by investigators including Robin Bell at the Lamont Doherty Earth Observatory of Columbia University, Fausto Ferraccioli at the British Antarctic Survey, and Michael Studinger at NASA. What they found was impossible. The Gamburtsev Mountains are approximately 1 billion years old. They formed during the Grenville orogeny, the same mountain-building event that created the core of the Appalachians. Mountains that old should not exist as mountains. A billion years of erosion, rain, wind, freeze-thaw, glacial grinding should have reduced them to flat peneplains. Yet here they were, sharp and rugged, looking like they had been carved yesterday. Bell and colleagues published the explanation in Nature in 2012. The mountains had been rejuvenated. The original billion-year-old roots, the deep crustal structures left behind after the original peaks eroded flat, were uplifted by rifting and tectonic forces between approximately 250 and 100 million years ago. This uplift raised the ancient roots high enough for rivers and then alpine glaciers to carve new peaks and valleys into the resurrected landscape. Then, the East Antarctic ice sheet advanced over the top of them, burying the mountains under kilometers of ice and freezing the topography in place. The two-phase model, Precambrian roots, Mesozoic and Cenozoic sculpting, Neogene burial. Beneath the mountains, the AGAP survey detected a network of subglacial lakes and water channels at the ice-bedrock interface, indicating active hydrological processes even under the deepest ice on the continent. The heat flow measurements from the region are unusually high for a crodon, a stable continental interior, suggesting enhanced geothermal activity possibly linked to the East Antarctic Rift system. That rift, mapped using AGAP data, shows structural similarities to the East African Rift, suggesting that Antarctica's interior is not the tectonically dead slab it was long assumed to be. The Gamburtsevs hold another prize. The basal ice above these mountains is estimated to be up to 1.5 million years old. The International Partnerships in Ice Core Sciences has identified this region as the most promising location on Earth for recovering ice older than 1 million years. An ice core spanning 1.5 million years would capture the mid-Pleistocene transition, the period between approximately 1.2 million and 700,000 years ago when Earth's glacial cycles shifted from 41,000-year periodicity to 100,000-year periodicity. Why that shift happened is one of the biggest unsolved questions in climate science. The answer may be frozen above a mountain range that no one can see. The ice sheet above the Gamburtsev Mountains is relatively flat. There is no surface expression whatsoever of the dramatic topography beneath. If the East Antarctic ice sheet melted entirely, a scenario modeled at atmospheric CO2 concentrations exceeding 1,000 parts per million, the Gamburtsev Mountains would emerge as a major alpine range surrounded by shallow seas, the total ice volume above them representing approximately 21.7 million cubic kilometers and roughly 58 meters of potential global sea level rise. They are the largest mountain range on Earth that no human has ever laid eyes on, and unless something goes catastrophically wrong with the climate, no one ever will. Number 10. The frozen rainforest roots. In 2017, an international team led by Johan Klages at the Alfred Wegener Institute sailed the research vessel Polarstern to the Amundsen Sea and drilled into the continental shelf near Pine Island Glacier at approximately 73 degrees south latitude. They were expecting sediment. They got a forest. The core, taken at a water depth of approximately 946 meters, recovered a layer of Cretaceous age sediment roughly 90 million years old from the Turonian stage, a period of extreme global warmth. When the team CT scanned the core back at the Alfred Wegener Institute, they found something that required them to recalibrate their understanding of what Antarctica has been. Preserved within the mudstone was a dense, tangled network of fossil roots, not scattered fragments, not transported debris. Roots in growth position, penetrating the fine-grained sediment exactly where they had grown, unmoved for 90 million years. Published in Nature in April 2020 under the title Temperate Rainforests Near the South Pole During Peak Cretaceous Warmth, the study assembled researchers from Germany, the United Kingdom, and Austria. Pollinological analysis, the study of pollen and spores, identified at least 65 distinct plant taxa in the sediment, including conifers, ferns, and early angiosperms. The plant assemblage most closely resembled the modern temperate rainforests of New Zealand's South Island, cool temperate, wet, and dominated by podocarps and tree ferns. The critical number is the paleolatitude. When these roots were alive, this site was located at approximately 82 degrees south, within 900 kilometers of the Cretaceous South Pole. This is the southernmost forest ever documented in the geological record. A temperate rainforest growing at a latitude where today the average temperature is negative 20 degrees Celsius and the land is buried under kilometers of ice. Climate modeling by the team estimated mean annual temperatures of approximately 12 degrees Celsius at this site during the Mid-Cretaceous, with summer temperatures reaching 18.5 degrees Celsius. Atmospheric CO2 levels were estimated at 1,000 to 1,600 parts per million, 4 to 6 times pre-industrial levels. At those concentrations, the greenhouse effect was powerful enough to eliminate polar ice entirely and maintain unfrozen ground year-round at 82 degrees south. The forest endured 4 months of continuous polar darkness every winter, no photosynthesis for a third of the year. The trees were adapted to a cycle that no modern forest on Earth experiences, months of nonstop growth in 24-hour summer sunlight followed by months of metabolic shutdown in total darkness. The Plesiosaur, the ancient soil, showed no evidence of permafrost. Biomarker analysis identified bacterial heapanoids and plant-derived terpenoids, indicating an active soil microbiome supporting the root network. Charcoal fragments in the sediment suggest fire was part of the ecosystem, likely ignited by lightning in a warm, seasonal climate. Precipitation estimates placed annual rainfall at approximately 1,100 to 1,200 millimeters, comparable to modern Seattle or London. The findings forced a reckoning with climate models. No existing model had predicted temperatures warm enough for rainforests at 82 degrees south, even at elevated CO2 levels. Klages and colleagues argued that cloud feedbacks and vegetation albedo effects had been systematically underestimated. A forest at 82 degrees south absorbs more solar radiation than bare ice or rockwood, creating a feedback loop that amplifies warming beyond what CO2 alone would produce. Modern Pine Island Glacier, which sits directly above these ancient forest sediments, is one of the fastest retreating glaciers in Antarctica, losing approximately 45 billion tons of ice per year. The Cretaceous rainforests beneath it were eventually replaced by cooler climate nothofagus forests as CO2 levels gradually declined through the late Cretaceous and early Cenozoic. The last forests in Antarctica survived until approximately 12 to 14 million years ago on the Antarctic Peninsula, before advancing ice sheets during the Middle Miocene eliminated them for good. The frozen rainforest roots beneath Pine Island Glacier are not just a curiosity, they are an empirical upper bound for climate sensitivity. They tell us, in the language of fossilized roots and pollen grains, exactly what happens when atmospheric carbon dioxide reaches 4 to 6 times pre-industrial levels. The polar ice vanishes, the glaciers melt, and forests grow where glaciers once stood, 900 kilometers from the South Pole, in soil that has not felt sunlight for 90 million years. Antarctica is not what it appears to be. The white continent, featureless, sterile, hostile, is a mask. Beneath the ice, the geological and biological record of this landmass is richer, stranger, and more disturbing than any other place on Earth. Pregnant marine reptiles frozen in the act of giving birth, glaciers that bleed iron, a lake the size of Ontario sealed in darkness for 15 million years, possibly harboring life. Spiders with 70-centimeter leg spans stalking a frozen seafloor. Forests still standing where they grew before the worst mass extinction in history. An entire microbial biosphere running on rock chemistry in permanent darkness. Barnacles recording whale migrations in their shells. A mountain range the size of the Alps that no human has ever seen. And the roots of a rainforest, 90 million years old, still threaded through the soil at 82 degrees south. Every layer of ice is a page. Every fossil is a sentence. Every microbe clinging to a subglacial rock face is a testament to life's refusal to accept the boundaries we assume are permanent. The continent's past tells us what happens when the planet warms and what grows in places we assumed were empty. The irony is that the archive teaching us these lessons is itself melting. The ice that preserves is the ice that is disappearing. And beneath it, the evidence of transformation, persistence, and deep time waits, as it is waited for millions of years, for someone to read it before it is gone.